Good morning, dear students. Today, in this section, we are going to start the topic Flagellar and Ciliary Moment in Protozoa. In the last few videos, we have seen the mode of infection pathogenicities of different protozoans. Today, in this section, we are going to see its different methods of movement. So many different methods are exist: swimming movement, amoeboid movement, and gliding movements. So each type we will see in this chapter. First, we are going to see about the swimming movement of protozoans. Swimming movement is facilitated with the help of flagella and cilia. First, we can see about the flagellar movement. For understanding the flagellar moment, first we need to understand its structure. So let's see the structure. The flagella, if we take a cross section of this flagella, we can see a 9 plus 2 arrangement. 9 plus 2 arrangement of microtubules inside. This is the cross section of the flagella. 9 plus 2 means as doubles, 9 set are there. And in the central, one more duplex. So let's count 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 duplex and one central separate duplex. That is the arrangement. Next, we need to understand about each microtubule. Let's take the case of one duplex of microtubule. This is one microtubule, and associate with that, along with that, there present another microtubule also. So this is one duplex. And among these duplex, one microtubule is complete. And the other one, it is incomplete. In what respect it is complete and the other one is incomplete? Complete means this microtubule is composed of 13 protofilaments. While the other one, it is composed of only 11 protofilaments. So while taking the structure, appearance wise, it looks like a complete circle, while the other one is the form of a letter C only. So that is the difference. That's what we need to understand about the each duplex of microtubule present in this flagellum. And between we can see some yellow portions we can see here. They are actually the dynein protein, its arms, dynein proteins. What is this dynein proteins? Dynein proteins are certain proteins that can catalyze ATP and can generate energy to facilitate the flagellar movement. I will show you an image of the dynein protein. This is a dynein protein, and it has a stalk, a head, and a tail. Using the tail, it carry the cargo or attach with something, and using the stalk, it is attaching with the other microtubules. So that's the structure. So in this picture, in very small size, we can see that dynein between the microtubule duplex. Okay, and one more thing, spokes means like the axial rays that we can see in a bicycle's wheels we can see such spokes. This is spokes, some more spokes. So similar to that, here also we can see spokes. So what's the purpose of spokes? To keep this duplex in position. So this is about the arrangement or internal structure of flagella. I hope that it is clear to you. So next we are going to see the mechanism of the flagellar movement, how it moves. For showing that, I will draw two microtubule duplex. First duplex and second duplex. So we know that in a circular fashion, this microtubule duplex are arranged in the flagella. Okay, so the other microtubule duplex I am not going to draw here, only two. So next thing we need to show here is the Dynein arm, Dynein. So Dynein's tail and its stalk is attaching with the nearby microtubule. Sorry. So this is the arrangement. This dynein protein, this is an ATP hydrolyzing protein. By the hydrolysis of ATP, it can generate energy, and using that energy, it can pull microtubule. So that's what I am going to show you. As it got energy, using the tail, it attached with the neighboring microtubule duplex and pull it backwards. As it pull this microtubule to backwards, while the time it pushes this microtubule duplex to forward. So this is the sliding type of movement that is happening inside the inside the flagellum between the microtubule duplex. And with the help of the sliding, that flagellar movement takes place. Then how that bending of flagella happens, how it bends. For understanding that, we need to learn that if we take the case of a flagellum as a whole, one side always getting the stimulating or promoting signal. Stimulating signal or promoting signal, that is allowing this microtubule duplex to move, while the other side is getting a restricting signal. That means the other microtubule duplex in this side cannot move. Its movement is restriction, its sliding is restricted, and because of that restriction, as a whole, that microtubule will bend. Okay, so after that, this signal alternate means the other side get the restriction signal, while this time gets stimulating signal. While because of that, then this side bend, this side can move and this side cannot move. So the direction of bending get alternated, and because of that, a whipping like this type of movement will happen or takes place. So I hope that is clear to you, and after showing this video to you, it will be more clear. So let's see, this is the Dynein using the tail. It is attaching to one duplex. Using the stalk, it is attaching with other duplex. And let's watch the way how it pulls this microtubule, and after that how it behaves when it get a restricting signal. So let's see. So it is pulling, and after that restriction signal, so it cannot attach with the microtubule, adjacent microtubule, inhibitory signal, so it cannot attach. So that means that sliding moment is absent in one side. So we are going to see the whole whole flagellum. So red is inhibitory and blue is stimulatory signal. So that signals are alternating, and that alternation, I can see that alternation alternately bend this that flagellum to two directions, to and fro. So we can see very clear restricting and supporting signals. It is alternating. So because of that, that direction alternates and create this type of movement. So that's about the movement of the mechanism that involved in the movement of flagellum. So let's see the steps. The flagellum pushes the fluid medium at right angles. I will draw here. Suppose this is a protozoan and this is its flagellum. And this is the surface where this flagellum attached with this protozoan. So we can take it. This is the surface. And a right angle to this surface. That means to in this direction be its movement. So that's what written here. Flagellum pushes the fluid medium at right angles to the surface of the attachment and also by its bending moment. How that bending occurs, I already explained to you. Let's see the steps. By the sliding of microtubules past each other with the help of Dynein arms happens that bending. And this Dynein arm, it show a cycle of its movement again and again to bring about the flagellar movement. Using what? Using the energy provided by the ATP. How that cycles happens, what are the process involved, that everything I have explained to you by this time itself. Anyway, once more, that Dynein arm attach the outer microtubule of adjacent doublet and pull the neighboring doublet, and because of that pulling they slide past in opposite direction. Okay, clear? And after that pulling, then this Dynein arm release from the attached point, and again it reattach the neighboring doublet to bring about another round of pulling. So the entire process cycles, and because of that pulling, that microtubule doublet, it will not slide past too much. Why? Because that sliding, the extent of sliding is restricted by the radial spokes that holding the microtubule doublets in position. So that's what. And as that sliding motion is restricted, then instead of too much sliding, it can only curve or bend the flagella. So this bending has an important role in flagellar movement. So I hope that's very clear to you how sliding occurs and how that sliding ends up in a bending only because of the help of radial spokes. So that's what. Then next we are going to see the types of flagellar movement. First one, undulation movement. Undulation movement itself are of two types. As pictures are shown here, in first type, that the protozoan exerts a force, pushing force towards the direction of water. That means that force is starting from the base of the flagellum towards the tip. And during that time, that protozoan is moving in opposite direction. This pushing that water and move back. We have learned a law. For each and every action, there will be an equal and opposite reaction in the same way. So that is the first type, undulation. And coming to the next one here, a pulling force. Pulling force means it is pulling water, taking water towards the direction of the attachment of its flagella. And during that time, that protozoan is moving forward. By pulling water towards it, it is moving forward. So that is the two types of undulation movement. Next is a sideways lash movement. Sideways lash movement is just similar to the wing stroke of bird or the paddle stroke of a boat. Means it is a sideways lash, consists of so many strokes, and that strokes are of two type: effective stroke and recovery stroke. First, we can see the effective stroke. Effective stroke, what is it? I will show the picture. Means the flagellum becomes straight, then it is striking against the water. By that way, the organism, it can move forward. So that is the effective stroke. Also, effective stroke means the flagellum becomes rigid first, and starts bending against the water and beat, and because of that beat the organism move forward. So that is effective stroke. Then recovery stroke means it is recovering from the effective stroke. Flagellum becomes soft and will less resistant to water, and because of that this flagellum move backwards to its original position. From this position it is moving back to its original position. That is the recovery stroke. I will show you. Recovery stroke, this one. While during the effective stroke, it is coming down to this direction, and after that, it is recovering by becoming soft and less resistance against the water. It is recovering or coming back to its original position. So I have tried here to generate that moment. So next one, simple conical gyration. Gyration movement means it's just like the how a screw turns through something. The screw movement. It is a propelling action, and because of that propelling action, it pulls the organism towards which direction? Towards the forward direction through the water with a spiral rotation around the axis of movement. Spiral rotation around the axis of movement means, suppose if that organism is moving, this is the axis of movement. Around that axis of movement, the flagellum shows a spiral movement, gyration. That is a gyration. Next, we are going to see the ciliary movement. Like the flagellum, cilium also show back and forth movement. That means effective and recovery strokes. Then what is the pattern? Pattern is, suppose this is the protozoan, and the movement of water is parallel to the point of attachment of the cilia. This is the point of attachment. So the movement of water that also parallel to the point of attachment of the cilia, and that movement is again it is perpendicular. It is perpendicular to the longitudinal axis. Longitudinal axis is this one. This is the longitudinal axis. So that moment is again perpendicular to the longitudinal axis of the cilium. In two ways, we are explaining the direction of movement of water by cilium. One, it is parallel to the point of attachment of cilium, but that moment is perpendicular to the axis, longitudinal axis of the cilium. Clear, simple. So that's the pattern of movement of water by the cilium. And again, I am explaining you about the two strokes, effective stroke and the recovery stroke, that we have already seen in the case of flagella. Here also, during the effective stroke, the cilium bends and beat against the water, and so the direction of propulsion of water is towards in this direction, and at the same time the organism moves in opposite direction. So that be the case always, means movement of the organism and propulsion of water be in opposite direction. So that is effective stroke. Effective stroke, the cilium bends against the water and move the body forward. Recovery means coming back to original position. Coming back to original position by the backward movement without any resistance, like the flagella itself. And then again, one more thing, I want to make it confirm that whether you know what is the exact difference between flagellum and cilium. Some differences are the cilia. I'm just writing cilia and flagella. The cilia are small, but flagellums are more usually they are longer than cilia, and also the cilia are numerous. Flagella most oftenly may be found either in anterior end or posterior, so they are a few in number. These are the some striking difference between cilia and flagella. And the cilia, while beating, it showed two types of rhythms, synchronous rhythm and metachronous rhythm. Synchronous rhythm means the cilia beat together simultaneously in a transverse row. While in the case of metachronous rhythm, all the cilia beat one another. Suppose this is the protozoan, all the cilia beat one another in a longitudinal row, and that wave pass from anterior to posterior, and that we can see here, metachronous type of beating. And while beating, whenever the cilia come across some obstacle, it can reverse its direction of beating and avoid that obstacle. That is another feature. Then how that coordination of that ciliary movement happens? It is with the help of a system called infraciliary system. What is infraciliary system that I am going to explain to you? This picture is shown here in these two pictures. Each cilium at its base present a kinetosome, and from that kinetosome emerge kinetodesmal fibrils, and all these fibrils then run towards the right side of each kinetosome as kinetodesmata, and this kinetodesmata together form one kinety. Okay, and all the kinetes from all the cilia together form that infraciliary system. And that infraciliary system is controlled, regulated, connected to a motorium, a controlling center called a neuromotor center, where it is located near to the cytopharynx. Cytopharynx, what I am going to show you, cytopharynx is this invagination like region. It is a tube-like opening that present in protozoans on its lateral surface, through where the food is ingested. So near to that cytopharynx, present that neuroregulator center or neuromotor center called the neuromotor system. It is regulating and controlling the entire beating of cilia. I am going to show that neuromotor system. This is that center. All the series, its beating is regulated and controlled at this center. Okay, that's all about that coordination. Again, the some more points. Points means I already have explained to you all the things. Once more, I am reading. This infraciliary system with the help of a neuromotor center called a motorium that I shown you. It is present near the cytopharynx. It is controlling the ciliary beating, and it is coordinating all the beating of the cilia in the case of protozoans. And this ciliary movement, it is the fastest locomotion in the case of protozoans. By this, we are stopping today's section. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.